Very good morning students, this is your Prasanna ma'am from NEET World. Now we are going to discuss about most important and interesting topics from principles of inheritance and variation. Those are chromosomal theory of inheritance and linkage and recombination. If we look into the previous concepts of this chapter, we have learnt about monohybrid cross, dihybrid cross and their principles. Okay, what are those principles that Mendel has derived from monohybrid and dihybrid cross? Those are law of dominance, law of segregation and law of independent assortment, and we have also discussed the exceptions of those principles, right? Now in this class we are going to learn about chromosomal theory of inheritance. These concepts arise because Mendel's laws are unnoticed or not recognized for a very long period of time. As we have discussed previously, Mendel has worked on Pisum sativum or pea plant for almost 7 years. Yes or no beta? For 7 years. Okay, and he has published all his work in the year 1865. But then his work is not approved or not come into picture up till early 20th century, that is up to 1900. His work has not come into picture because of many reasons. Let us look into what are those reasons because of which Mendel's laws are not recognized or Mendel's work is not approved. The very first thing is limited communication. Those days the scientists have very limited scientific communication and their work is not much published. Second most feature is opposition of discrete inheritance or non-blending inheritance, or what beta? Non-blending inheritance. Okay, which means at that time the concept of evolution or variation is very much popular, so that the concept of genes or factors which are transmitting from one generation to other generation without any mix or blending of characters, without any mix of the alleles, they are transferred from one generation to other generation, right? This is what explained by Mendel. That is not approved because at that time variation and evolution is more popular. This is contradictory to this statement. The non-blending or mixing of alleles, and transmitting from one generation to other generation is contradictory to the theory of variation from evolution. So that is the reason this is not accepted. And third is Mendel's mathematical and statistical approach. Okay, his mathematical and statistic approach didn't fit into those minds. Okay, biology involving statistical and mathematical approach was quite a new approach at that time. So that is why this is not approved again. And lastly, lack of physical evidence of the genes. Yes, it is very good that Mendel has highlighted about factors which are in the chromosomes. Okay, he has highlighted that factors are transmitting from one generation to other generation without any mix, and this is how the pattern of inheritance is floating out. Yes, but then there is no physical evidence, there is no physical proof to prove those statements, and that is the reason it is not accepted. So yes, the most unfortunate scientist we will say it is Mendel, because his work is not recognized in his tenure. Okay, then how and when is his work recognized? His work got recognized because of these three scientists. Those are De Vries, Correns and von Schermach. Okay, who are those three scientists beta? De Vries, Correns and von Schermach, who independently or individually proved the Mendelian principles. They individually or independently proved Mendelian's principles. So yes, in 1902 or in the early 20th century, the discovery of microscope has also played a crucial role in understanding the cell biology, in understanding the cell cycle, in understanding the pattern of chromosomal segregation. Okay, so let us see how the chromosomal segregation happens. Of course, we have discussed this in the cell cycle, but then once let us recall it again. So if you look here beta, in G1 phase, of course in cell cycle we have two phases, that is interphase and M phase. When we are talking about chromosomal segregation, it is meiosis in the M phase. It is meiosis that occurs in the M phase. In G1 phase, the chromosomes which are present, DNA which is present in the chromosomes is duplicated or replicated in S phase. DNA gets duplicated or replicated in the S phase. Chromosome number remains same, this we know. So in S phase what is happening ma? Chromosomal duplication happens. After G1 it is S phase. In S phase chromosomal duplication happens. What happens ma? Sorry, what happens? DNA duplication. DNA duplication or replication of DNA occurs. Clear beta? And after G2 phase, the very first phase takes place in meiosis is prophase 1. What happens in meiosis after G2 phase? Prophase 1. We know in prophase 1 we have again 5 phases. What are they, those 5 stages? Leptotene, zygotene, pachytene, diplotene and diakinesis. Among all these stages, in which stage does recombination occurs? In prophase 1, it is in the pachytene stage. It is in the pachytene stage the recombination occurs. So the traits gets exchanged. The traits gets exchanged or recombined in pachytene. The traits gets exchanged or recombined in which phase? Pachytene. Okay. So after exchanging of the genetic information or after recombination, then diplotene diakinesis, after then metaphase 1 occurs. In metaphase 1, the pair of chromosomes are aligned in a bimetaphasic plate. Okay, pair of chromosomes are aligned in what beta? Bimetaphasic plate, in two metaphasic plates. And in anaphase 1, chromosomal segregation happens. Chromosomal segregation happens in which phase? Anaphase 1, wherein these two chromosomes, when there are set of chromosomes, these two chromosomes will move towards the opposite pole without any centromere splitting. The sister chromatids of each chromosome will move towards the opposite poles. And in the anaphase 2, this chromosome, of course this sets into one cell, this sets into other cell. Here in the anaphase 2, centromere splitting occurs. Centromere splitting occurs, wherein two chromosomes get separated and they form half the set of chromosome. We will call it as haploid chromosomes. Okay. So in the early 20th century what happened beta? These three scientists, that is De Vries, Correns and von Schermach, has proved the Mendelian's laws of inheritance or Mendelian's discoveries independently or individually, and then due to the discovery of microscope, vast amount of research has gone in cell biology or understanding the cell cycle or chromosomal pattern of segregation, wherein it is proved that in S phase DNA duplication happens, chromosome amount remains the same, DNA duplication or replication happens in S phase, and exchange of genetic information, exchange of alleles or traits happen in the pachytene stage of prophase 1. In metaphase 1, bimetaphasic plate is formed which is transferred to anaphase 1. In anaphase 1, two sister chromatids move towards the opposite pole without any centromere splitting, most important. Okay. And in anaphase 2, centromere splitting happens, which leads to the formation of 4 haploid chromosomes, which lead to the formation of what ma? 4 haploid chromosomes or 4 haploid daughter cells. This is what happens in the cell cycle. Now, it is the scientist who is Sutton and Boveri. Who is he? Sutton and Boveri has proved that chromosomal segregation and Mendelian pattern of inheritance. What did he do beta? he has combined chromosomal segregation with Mendelian pattern of inheritance. Chromosomal segregation is combined with Mendel's pattern of inheritance. Mendel's pattern of inheritance, and he has set some concept called as chromosomal theory of inheritance, wherein it is said that chromosomes occur in pairs. Chromosomes and genes both occur in pairs. Just a second. Yes, chromosomes or genes occur in pairs, as we know, right? For example, if we take a chromosome, where are genes present in the chromosomes, beta? Genes are present on the chromosomes. Yes or no? Yes. So, if we take any trait, for example, round and wrinkled, yellow and green, okay. So, genes are present on the chromosome at the homologous loci, on the same location, okay. So, chromosomes occur in pairs and genes also occur in pairs. During the time of segregation, one pair of the chromosome will segregate and one pair of the gene is also segregating independent to each other. This is what said by Sutton and Boveri. By uniting the concept of chromosomal segregation and Mendelian pattern of inheritance, he said that chromosomes occur in pairs and genes also occur in pairs. Segregation occurs during the time of gamete formation. Here also segregation occurs during the time of gamete formation, and here independent pairs segregate independently to each other. Here also one pair segregates independent of the other pair. In a gene, alleles, one pair will segregate independent to the other pair, just now we have seen. So, yes, we will understand this with a example in a better way. If you take Mendelian dihybrid cross, beta. In Mendelian dihybrid cross, what are the two traits we have taken? Two traits we have considered in the Mendelian dihybrid cross is seed shape and seed color. And what ma? Seed color. Seed shape, the dominant trait is round. So, the notation is given with respect to the dominant trait only, round, and small alphabet we will use it for the wrinkled. Keep it in mind, beta. And seed color, dominant yellow and recessive green. Clear? So, these two traits are considered for understanding the Mendelian's pattern of inheritance. So, in F1 generation, if we take in F1 generation, when we have this set of cells, okay, when we have heterozygous set of cells wherein we have round and wrinkled, yellow and green. Okay, round and wrinkled, yellow and green. When they undergo self fertilization or self cross takes place, there are two possibilities of having gametes. Okay, what are those two possibilities? We will understand now. Let us consider this as round. This chromosome or the genes which are present in this chromosome will carry a trait that is round, and this is wrinkled. Let us assume this as green and this as yellow. Okay, during meiosis 2, what happens? Centromere splitting happens directly from anaphase 1. In anaphase 1, this is the scenario wherein this is one cell now, this is the other cell after anaphase 1. Okay, now in anaphase 2, here centromere splitting happens, and we have 4 set of gametes here. What are they, beta? Here it is round and green, here again it is round and green, here it is wrinkled and yellow, here it is again wrinkled and yellow. Here if you see again, if we take two traits, that is round and wrinkled, and let us exchange the position of those chromosomes which are giving the traits of other, like yellow here and green over here. Let us exchange the positions of these chromosomes which are having the trait of seed color. Okay. Now if we see, here it is a round and yellow, and after anaphase 1 this will be one cell, this will be one cell, right. So here it is again wrinkled and green. Now after anaphase 2 what happens now? After anaphase 2, here what set of gametes are formed? The set of gametes are as such round and yellow. Here again it is round and yellow. Here it is wrinkled and green. Again here it is wrinkled and green. So at the end of F1 phase, to conduct the crossing and to analyze all the possibilities of zygotes in the F2 generation, we need to first know about all the possible gametes which are involved in the crossing. Yes or no? So when we look into all the possibilities, possibility 1 and possibility 2, we will have 4 set of gametes. How many set of gametes beta? 4 set of gametes, round and wrinkled. Second set is wrinkled and yellow. Third set of gamete is round and yellow. Fourth set of gamete is wrinkled and green. Fourth set of gamete is what ma? Wrinkled and green. These are the 4 set of gametes which are involved in the fertilization, and cross between these 4 gametes of male and female will give you how many? 16 zygotes. How many zygotes? 16 zygotes. Okay. How you said ma'am it is 16 zygotes? Gamete square will give you number of zygotes. 4 square it is 16. Clear ma? Next, if you see, yes. Who discovered chromosomal theory of inheritance? Just now we have discussed. It is Sutton and Boveri. Remember these names for sure. It has asked multiple times in the NEET, both in the NEET and EAP set. Okay, who discovered chromosomal theory of inheritance? It is Sutton and Boveri. And on what basis they gave chromosomal theory of inheritance? They have united the chromosomal segregation and they have united the Mendelian's principles. Okay, putting these 2 laws combinedly, they have said that chromosomal theory of inheritance. Okay, this is what we have discussed about chromosomal theory of inheritance. Who gave it ma? Sutton and Boveri. On what basis did they give? Chromosomal segregation and Mendelian principles. The pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. Okay, just now we have discussed. Factors are present on the chromosomes. Factors are present on the chromosomes. Segregation of the chromosomes will lead to the segregation of factors which are present on the chromosomes. Okay, clear? Now we also need some experimental proof for the chromosomal theory of inheritance, right? So the experimental proof for chromosomal theory of inheritance is given by Thomas Hunt Morgan. Okay, it is given by Morgan, who is said to be the father of experimental genetics. He is said to be the father of experimental genetics. And what is the model organism he took? He took the model organism Drosophila. Okay, who gave chromosomal theory of inheritance? Sutton and Boveri. And who gave experimental proof for chromosomal theory of inheritance? It is Morgan. Okay, on what did he conduct the experiments to prove the chromosomal theory of inheritance? Drosophila. Okay, and in the process of conducting the experiments, the results, based on the results of the experiment, he has also gave us different concepts like linkage, recombination or crossing over, sex linkage and crisscross pattern of inheritance. All these discoveries he made based on the results of the experiments conducted to prove chromosomal theory of inheritance. He wanted to prove this theory, but then the results gave us the different phenomenons. Okay, that led to the discoveries of linkage, crossing over, sex linkage, crisscross inheritance. Never get confused whenever this question is asked. Who discovered linkage, crossing over, sex linkage and crisscross inheritance? Your answer should be Morgan. Who gave chromosomal theory of inheritance? Your answer should be Sutton and Boveri. Clear ma? Next. Who rediscovered Mendelian pattern of inheritance? Then what will be your answer beta? De Vries, Correns and von Schermach. Okay. Yes. He is also known as flyman of genetics because he has conducted all his experiments in Drosophila melanogaster, which is also said to be what? Fruit fly. It is also said to be fruit fly. Clear beta? Now if you see Drosophila male and female. Why did he choose Drosophila? We have discussed, Mendel's model organism is pea plant, Pisum sativum. Why pea plant? We have discussed many reasons why Mendel has chosen pea plant. Now there are many reasons why Morgan has chosen Drosophila as his model organism. First thing is, it is easy to grow on the culture. It is easy to grow on the culture because naturally it is easily grown on fruit surfaces, okay, and the time to generate new generation or its mating time and to generate a new generation, it will just take 2 weeks, okay. The Drosophila will just take 2 weeks in giving out or producing out the new generation. And one more advantage is, single mating of Drosophila will produce hundreds of individuals. Single mating of Drosophila will produce what ma? Hundreds of individuals. Second thing, females are easily distinguishable than males because they have large ovipositor. Because they have large ovipositor, females are easily distinguished when compared to males. Just by looking into hand lengths, we can distinguish between male Drosophila and female Drosophila. And in Drosophila, the sex determination will follow which heterogamety beta? They follow XY male heterogamety, okay. Males of Drosophila will have only one X chromosome and one Y chromosome, okay. Females will have 2 X chromosome, wherein it is following what pattern of sex determination? Male heterogamety. Based on this also, it is easy to analyze heteromorphic chromosomes. It is easy to analyze heteromorphic chromosomes that are being transferred from one generation to other generation. Keeping all these things in mind, Mendel has chosen the model organism to conduct the experiments as Drosophila. Clear? Now, what experiment did he conduct and what traits did he take? Mendel also conducted dihybrid cross only beta. What cross did he conduct? Dihybrid cross. Yes, dihybrid cross. Okay, but 2 sets of dihybrid cross he has conducted. One is cross A and the other is cross B. What are the traits he has assumed? He has taken 2 traits. He has taken 2 traits. Let us take one female and one male chromosome. Okay, this is XX. Just a second. Yes, this is XX and this is XY. Okay, he has taken how many traits? 3 traits into consideration. What are those 3 traits beta? One is body color and the other is eye color, and third character is wing shape. Third is what, Nana? Wing shape. Okay, so first if you consider body color can be red and yellow. It can be red and yellow. Okay, sorry, it can be brown and yellow. It can be what, ma? Brown and yellow. If you take eye color, it can be red and white. It can be what? Red and white. If you take wing shape can be miniature wing, miniature and normal wing. Now you have to know which is dominant and which is recessive trait in this. Okay, which is dominant and which is recessive. When you are taking body color, brown body color is the dominant trait. Okay, when you are taking eye color, red eye color of Drosophila is the dominant trait. When you consider wing shape, normal wing shape is the dominant trait. In Mendel, if you observe, the alphabet or the notion has given with respect to dominant trait, okay. But if you consider Morgan experiments, the notion or the alphabet is given with respect to recessive trait. For example, body color, if it is Y, it is recessive. If it is Y plus, it is dominant. Understood beta? If it is eye color, white it is recessive, W plus it is red, okay, dominant eye color. If you take wing shape, miniature is recessive trait, no, so M, and dominant is M plus, normal wing is M plus. This is what the notions that Morgan has given. Once you get to know about this concept, then only you will understand this diagram, okay. If you want, I will repeat once again. Let us take one allosome of female and this is male, XX and XY, okay. 3 traits he has considered to conduct 2 crosses. What are they, ma? Body color, eye color and wing shape. If you take body color, brown and yellow are the 2 body colors and the dominant is brown. Eye color, red and white, dominant is red. Wing shape, miniature and normal, and the dominant is normal. The alphabet or notion to indicate the trait is given with respect to recessive trait, okay. It is given with respect to recessive trait. For example, Y, when you are considering body color it is Y and brown it is Y plus, okay. Eye color it is W and dominant it is W plus. Wing shape it is miniature is recessive, so M, and normal wing it is M plus, okay. Now you have to understand one more thing before getting into the understanding of cross A and cross B. One thing, the position of the genes or the alleles, as I said you, is always on the same location, homologous, okay, homologous loci. Let us assume the body color and eye color are close to each other. Body color and eye color are close to each other on the same chromosome. Whereas wing shape trait, okay, these alleles are far from each other, okay, these alleles or this gene is far from each other. This is the approximate position of the traits on the chromosomes according to the Morgan. Now you look into the cross A. If you see here on chromosome, on the chromosome female, X and X, this is X and Y, okay. Here yellow and W, and here it is yellow body color and white eye color. They are close to each other on the same chromosome. Yes or no? In female, yellow and white, these 2 traits are close to each other on the same chromosome. If you take male also, brown and red, okay, brown body color and eye color, that is red, they are present close to each other on the X chromosome. And do remember, Mendel has explained only about X-linked inheritance. His crosses are based on what, beta? X-linked inheritance. Okay, now in F1 generation, if you see, okay, F1 generation, that parental generation, and in F1 generation, these chromosomes are close to each other and this is the pattern of inheritance. So in F1 generation you got 2 chromosomes, one with yellow and white and the other one is brown and red. In F2 generation, if you see, okay, after the cross if you see the F2 generation, or if you see the gametes of F1 generation, the gametes of F1 generation has 98.7% of parental combinations. They have what, ma? 98.7% of parental combination in cross A and just 1.3% of recombinants in the gametes of F1 generation. Okay, now these gametes will conduct the cross of F2 generation. Clear, ma? So here, as the genes are arranged close to each other on the same chromosome, it is you can understand it very simply. The genes are present on the same chromosome and they are closely linked to each other, closely attached to each other. That is the reason the parental combinations are more and the recombinant combinations or the recombinant type is very minimal. Okay. If you look into cross B, in cross B, these 2 characters are considered, one is wing shape and the other is eye color, wing shape and eye color. They are also on the same chromosome, but then they are not tightly linked, they are loosely linked, they are arranged far from each other, because of which crossing over happened, recombination happens, and then the recombinant type is 37.2%. The recombination percent is 37.2%. Now here, after conducting this crosses, it is not matching with the ratio of 9:3:3:1 which Mendel has gave us. Okay, it is not matching to this. It is because the genes are closely linked, or the position of genes is close to each other on the same chromosome. The position of genes is close to each other on the same chromosome. Here, when it is tightly linked, the recombination percentage is 1.3. Here, when it is loosely linked, the recombination percentage is 37.2. Recombination percentage is also said as non-parental combination. Okay, whenever in your question they are asking you body color and eye color, what is the non-parental combination? What is the non-parental combination gamete percentage? If the question is as such, then your answer is 1.3%. Okay, and in the diagram, if they are giving wing shape and eye color, where they are far from each other on the same chromosome, it is loosely linked, right? It is loosely linked. That is why non-parental combination or percentage is higher when compared to cross A, that is 37.2. You have to fix it in your mind. I hope this is very much clear, right? Now here comes the concept of linkage and recombination. The physical association of two genes on a chromosome. Okay, physical association of two genes on a chromosome. Just now we have discussed. Okay, one is body color and other is eye color, and the last one is wing shape. Okay. The physical association of two genes, on the same chromosome or on one chromosome is said to be what ma? Linkage. And the term recombination has come to describe the generation of non-parental gene combinations. This will give you what beta? The generation of non-parental gene combinations. Okay. This happens when two genes are far from each other and genes which are present on two different chromosomes. And you have to remember this point, which is maximum and most of the times asked, maximum recombination percentage. Maximum recombination percentage will be 50. Maximum recombination percentage is 50%. It is what ma? 50%, not 100%. Maximum recombination percentage is what beta? 50%. Now, that is how we are variant from our parents. If it is complete, then it will not happen at all. Okay. So, yes, maximum recombination is 50%. Morgan and his group also found that even when genes were grouped on the same chromosome, if they are tightly linked, as we have seen in cross 1, as we have seen in cross A, if they are tightly linked, the recombination percentage will be 1.3. Okay, just 1% or 1.3%. If they are loosely linked, it is around how much percent beta? If they are loosely linked, 37, it is around 37.2%. It is around how much percentage? 37.2%. Clear? Now, two genes will show higher frequency of crossing over when they are far from each other. That is what we have learned just now, no? Two genes will show maximum recombination. They will show maximum recombination. For example, eye color and wing shape. They show maximum recombination when they are far from each other. So, here we can say that distance is directly proportional to recombination. Distance is directly proportional to recombination or crossing over. Distance is directly proportional to recombination or crossing over. Clear beta? So, here if the distance between the genes is more, then there is more recombination and crossover. If the distance between genes is less, as we have seen between body color and eye color, as we have seen between what ma? Body color and eye color. Okay, this is W and W plus, Y and Y plus. Yeah, as we have seen between body color and eye color, if the distance between the genes is less, recombination or crossover percentage will be less. Clear? You have understood, right, the concept of recombination and linkage. With the concept of linkage and recombination, there is one more scientist, that is Alfred Sturtevant, who said about chromosomal mapping. Who said about what, ma? Chromosomal mapping. This concept deals with the position or the distance of the genes in the chromosome. This concept deals with what, beta? Position or distance between genes or the chromosomes. Between genes on the chromosomes. Okay. For example, there are 5 genes present on this chromosome. Okay. He might be giving you the distance between A and E as 15 as 15 centimorgans. Okay. The units is what? Centimorgan. And he might be giving you the distance between A and B as 10 centimorgan and B and D as 30, and he might be asking you the distance between B and C, distance between B and C. This might be the pattern of question. So what you have to do here, this also let us assume C and E distance is 20 centimorgan. Okay, yes, C and D is 20, he is asking you B and C. From the complete length of genes, first you have to choose the extreme genes which are there in the chromosome. Let us say this A and E are the extreme position, right? So from 50 you have to take and then combine, add on the distance between these 2 genes and these 2 genes which he has provided. Okay, let us say here 10 he has given, right? Plus you add on this 2, it is 20. So 50 minus 30, right? 50 minus 30, it is 20 centimorgan. Okay, it is 20 centimorgan. The distance between B and C is 20 centimorgan. Okay, this might be the pattern of questions. Okay, so who discovered chromosomal mapping? Alfred Sturtevant, and he has first given the chromosomal map from Drosophila only. Okay, and to calculate the recombination frequency also, just now we have discussed about recombination frequency of gametes from F1 generation, right? So to calculate the recombination frequency, there is a formula: total number of recombinants by total number of progenies into 100. Okay, what is that formula? Total number of recombinants by total number of progenies into 100. Okay. The recombination frequency always depends on the distance between 2 genes. Distance between 2 genes is more, then the recombination frequency will be more. Distance between the 2 genes is less, recombination will be less, and linkage will be more. Okay, linkage will be more. Okay, this is what you have to see. If recombination frequency is 5%, if the answer we got, let us say after calculating this, the recombination frequency is 5%, then the distance between the genes of that chromosome is 5 map units. How you will calculate between the distance of 2 genes? In map units and centimorgan. Okay, clear? Clear with this beta, and this formula you have to keep it in your mind. Of course, we will exercise all the applications of this formula in our questionnaire session. Don't miss watching the questionnaire sessions. Those are very, very important. Okay. Any doubts in this session, please drop in the comment section. I will be answering all your doubts. Thank you.